We observe heritable variations among individuals in a population, and these traits are transmitted from parents to offspring. One possible explanation for heredity was a blending hypothesis that proposed that genetic material contributed by each parent mixes in a manner analogous to the blending of paints. Everyday observations and the results of breeding experiments tell us that these traits don't actually blend that way, with Mendel being the father of this overall study. Mendel started his experiment with varieties of peas that were so-called true breeding peas. And true breeding plants self-pollinate. All of their offspring have the same traits as their parents. In a typical breeding experiment, Mendel would cross-pollinate two contrasting true breeding pea varieties, and he would look at the results of the pea generation and the supplement filial generations, F1, F2, and so on. Overall, it was mainly Mendel's quantitative analysis of the F2 plants that led him to deduce the fundamental principles of heredity, segregation, and independent assortment. Overall, we know now, using modern terms, that traits come in alternate versions or alleles, and they vary in nucleotide sequence at specific gene loci or locus. Traits are inherited as discrete units that we call chromosomes, with two alleles for each trait being a diploid organism, one allele from each parent. We know that some traits mask others, some traits are dominant, some traits are recessive, and Mendel believed, at least, that genes were not linked in any way. So either the genes are on separate chromosomes or far away from each other. Those are, in fact, unlinked. Mendel was not right about everything, which we will look, but that did introduce the basic principles that we know now. Overall, though, Mendel got really lucky with his peas. They were available in many varieties with distinct heritable features with different variations. He had strict control over which plants mated with which. He could cross-pollinate them. They can self-fertilize. They're relatively simple genetically. So there's a lot of advantage here. There are many traits that are a simple yes, no. That's not really something that we see with most human traits, for example. He avoided traits that existed on a continuum or traits that varied. So he got very lucky with the 7 characters that he studied. We know now that these 7 characters were in fact unlinked, which means, like we said a second ago, they're either very far apart on the same chromosome or they're on completely different chromosomes. Other studies that had attempted to do this, like Darwin attempted to do this, but he's tried to do it with roses, which were much more complicated genetically. Overall, reviewing a couple of those terms, you have the phenotype, or the physical expression, and you have the genotype, the genetic makeup. The genotype is going to be different alleles, different sequences of DNA that lead to different products. In this example, we have the purple gene and the white gene. The purple gene produces a functional enzyme that will produce pigment, whereas the white gene produces no enzyme and therefore no pigment. This is why the purple gene is dominant over the white gene. We study these things by using Punnett squares. This is obviously something that was not invented by Mendel. It was invented by a man named Punnett, but they are a useful tool for studying these, and so we'll use those more as we go in. Just try to understand that those basic terminologies. It gets more complicated over time, but those terms are nonetheless true. Another way that we can study the interrelationship between organisms is looking at pedigree charts. These are going to be constructions that show inheritance patterns over multiple generations, and you'll need to be able to interpret them. They will not always be labeled with the allele versions, but we should know that the capital letter typically represents a dominant trait. The lowercase letter typically represents a recessive trait. You typically are going to keep the dominant letters first, and you keep the traits together, which we'll try more in class. Squares represent males, circles represent females, and lines represent descent and connection. If you try to understand different pedigrees, here's some examples. We can find that there are many different types of patterns and structures that can exist within the organism. The first one that we see on the top left is what's called an X-linked recessive trait. You might notice that the trait mostly affects males because males only have one X chromosome. They are hemizygous for the X. Any male that inherits an X-linked recessive disease will be affected by it. Females will need 2 copies. color blindness, hemophilia, red-white eye color in fruit flies, for example. Mitochondrial DNA inheritance, you can see an example of that on the right. It's inherited through the maternal line from your mother. This is because although everyone has mitochondrial DNA, the sperm does not carry any mitochondrial DNA to the egg during fertilization. You can identify a mitochondrial DNA inheritance by noticing all of the siblings in a generation show the trait, yet only females pass it on. They don't always have to show it, it gets more complicated, but if you look at a trait and it's only passed on by a female, then you need to look at something like mitochondria, or if it's a plant, maybe a chloroplast, passing that trait along. The example on the bottom is an example of a Y-linked trait. Men have Y chromosomes, females typically do not, if you are at least in the XXXY sex-determining system like humans. Only the father to son is transmitting it, so it's an easy mode of inheritance to identify. Just look for all of the males in a particular lineage showing a trait. For example, there is a hairy ear rim phenotype that's seen in some family lines that is a Y-linked trait. If you combine some of these things together with the Punnett square and trying to figure out those unknowns, you can end up with what's called a test cross. A test cross is a very useful tool when you have a dominant, but you're not sure if it's a heterozygous or a homozygous dominant. If it's a homozygous dominant and you cross it with a fully recessive organism, you would get all purple offspring in this example. You would get all dominant. If it was a heterozygous, you would get some that were not dominant. So a test cross can be a useful tool for studying that. And this ties into one of Mendel's observations, which would be segregation. So when gametes are produced during meiosis, homologous chromosomes separate from each other. Each allele for a trait is segregated into separate gametes. And so that law of segregation, which has exceptions, so it's more a principle, basically just says that two alleles for heritable characters segregate during gamete production and end up in different gametes. This segregation corresponds to the distribution of homologous chromosomes, and it's also why Punnett squares work. Mendel's other observation, independent assortment, is a little bit more complicated, and it applies to more than one gene at a time. Each pair of alleles segregates into gametes individually, and it's only true for genes on separate chromosomes. When you're looking at this sort of broader relationship, you need to first realize that you have to produce the gametes. In this example, the male and the female, or the two P's, are only able to produce one gamete each, so the F1 generation will all be heterozygous for both traits. If the traits were dependent, if they were linked, it would act like a normal Punnett square, a monohybrid cross with that 3-to-1 relationship. If the traits are unlinked, if they will segregate and independently assort, then you're going to end up with a much more complicated arrangement, which we call independent assortment. For a fully heterozygous cross of independently assorted unlinked genes, you would end up with that phenotypic ratio of indicative 9:3 to 3:1, with 9 being dominant for both traits, 3 being dominant for one trait but not the other, and vice versa, and 1 being recessive for both. So Mendel's independent assortment says that each pair of alleles segregates independently during gamete formation, which is what we see here. So on the left, the gametes stayed the same as the gametes of the P generation, but for the ones on the right, the gametes are different. This really also then ties into some more complicated structures. So once you have this understanding, you don't really need to do Punnett squares for trihybrid or pentahybrid or whatever. You just need to apply the rules of multiplication and the rules of addition. The rule of addition says that there's a chance that an event can occur 2 or more different ways. If that's true, you can add it up. The rule of multiplication says that the chance that 2 or more independent events will occur together can be computed by multiplying the individual probabilities. So with Mendelian genetics, you can do this same cross that we see on the left with some simple math. If you take the 2 gamete combinations for the first and the second, you then can just multiply those relationships. So we can find out that there is a 1/16th chance of producing fully homozygous recessive, which relates to that 9:3 to 3:1. If you add up the things on the right, you'll get that same relationship. You don't have to apply this to only heterozygous crosses. What you would just do is you would take the trait, put it on a simple monohybrid relationship, and then multiply. We'll do some examples of this in class. Overall, though, I said that Mendel wasn't right about everything. We know a lot more now than we did before. Dominance, for example, is not an absolute trait. Many inherited traits result from modes of inheritance that differ from a strict dominant recessive relationship. Phenotypes can result from alleles with a range of dominance, from the combined effects of more than one group, or from genes that have more than just two alleles. Incomplete dominance, for example, is going to be when there is a condition where the dominant allele does not completely mask the recessive. As a result, some of the recessive trait will be observed in the phenotype. An example of an incomplete dominance may be colors of flowers. Red is dominant over the recessive color white, so pink flowers are going to be a result of incomplete dominance, blending the two. Codominance is going to be a little different. So codominance means that there are traits that are going to be equally expressed, which we can see within blood type, for example. So if you have AB blood, then you're going to have both the A and the B antigens on the surface. If you have A blood, you only have A antigens. If you have B blood, you only have B antigens. If you have O blood, you have no antigens, because it actually was a 0 at first. Okay. So codominance with flowers would look more like on the top, the red and the white, with incomplete dominance being a pink. So blood type is an example of one where you have codominance, but you also have multiple alleles. There are 3 possible alleles for blood type, but we only inherit them in pairs because we are diploid organisms. So dominance is clearly not an absolute characteristic. Another complication is what's called epistasis. Epistasis is when 2 or more genes control the expression of a single trait. In this example, you have the ability to produce pigment, which is controlled by the E allele, and the color of the pigment determined by the B allele. So you end up with that fully heterozygous cross producing a 9:3:4 relationship in this example, because even though the golden labs should have black or brown pigments, because they have 2 little e's, they don't produce any pigment. So we have an alteration that is an epistatic trait, with one trait preventing the expression of another. It doesn't just apply to dogs, it applies to lots of things. But if you have a heterozygous epistatic cross of a dihybrid trait, 9:3:4 is that indicative relationship. Other traits are polygenic. These are going to be traits that are controlled by 2 or more genes. These traits show a great variety of phenotypes like skin color and height and eye color, things like that. Having all dominant alleles results in, say, black eye color, where the presence of at least 2 dominant alleles produces the black or brown color. The presence of one dominant allele produces the green, while having no dominant alleles produces blue. Skin color is another example, with 6 different genes controlling skin structure. So if you look at a trait and it exists on this continuum, on this bell curve, it's probably a polygenic inheritance. So some characteristics cannot be classified as an either or, like Mendel's were, and we need to realize that it gets a little bit more complicated. Another factor that plays a role is the environment. Human body weight, for example, is controlled by dozens of factors, not just genetics. The bunnies on the left are the same species. The pigment breaks down in winter, so the white rabbit has lost its pigment, not grown out new fur. The hydrangea flowers are being planted in different acidities of soil, producing different flower colors. So we apply this to humans, and compiled twin studies from around the planet suggest that we need to not look at ourselves as nature versus nurture, but instead to realize that we are a combination of both nature and nurture. Phenotype depends on both environment and genes. Okay, nutrition, for example, will influence your height. Exercise alters build. Sun tanning darkens skin. Experience improves performance on intelligence tests. There are multifactorial characters where the environment has an influence just like genes do. We also have the additional complication of inherited disorders. Inherited disorders are going to be widely complicated, but it gets even worse when you look at traits that express what's called pleiotropy. Pleiotropy is when one gene has multiple effects on the physiology of an organism. We find out that many genes are pleiotropic, especially most inherited disorders. So pleiotropic affects more than one phenotypic characteristic. It's kind of the opposite of polygenic. Polygenic was many genes, one trait. Pleiotropy is many traits from one gene. And considering the intricate molecular and cellular interactions responsible for development, it's not surprising that a single gene can affect a number of characteristics. The last sort of complexity here is that some of these traits that exist, some of these inherited disorders, also differ in what's called penetrance and expressivity. Okay, expressivity is when you have the variation in phenotypic expression when an allele is penetrant, whereas penetrant is the likelihood of a gene expressing a phenotype. Expressivity is the extent to which the trait is expressed. They've been defined through clinical experience, but it really gets complicated. Okay, a genotype which may or may not produce a clinical trait is said to be incompletely penetrant. Okay, and then the extent, that variable expressivity, is going to be the range of symptoms that can occur in different people with the same genetic relationship. Okay, if penetrance varies, genotypes sometimes do not penetrate through the phenotype. Okay, so lack of penetrance can be used to explain individuals who do not reflect their inferred genotypes, and that's when you have traits that skip a generation. If expressivity varies, then the genotype will not be expressed uniformly in the phenotype. Okay, so we get that idea there. If expressivity and penetrance both vary, there may be a continuous gradient of phenotypes between the structures. Okay, lack of penetrance can be extreme, and it can be as an extreme form of expressivity, so it gets really complicated here. So really you need to understand more about the individual disease that you are studying, and just because a person has the gene doesn't mean it's going to express the trait, and realize that while Mendel's work is fundamental, it inherently does not take in the complexity of genetics that we are currently understanding, as well as the complexity that we have yet to understand.